I'm excited to be able to talk about our work. Before I get started, I'd like to introduce my partner, Ned Ruby. Ned, can you raise your hand? Yeah, so I'm going to be talking about a symbiosis today. And I am very animal-centric because I'm the person who studies the animal side. And Ned studies the luminous bacteria that are associated. And so if there are questions and they are of a bacterial nature, I'll probably turn them over to Ned, Dr. Ruby. So what I learned because of some technological advances in molecular biology in the last 10, 20 years is that animals require microbes for their health. And almost all animals, if not all animals, live persistently with microbes. They grew up, they evolved in a rich microbial soup, and they came to rely on them. And we couldn't know that before because we couldn't identify the microbes. And now we're able to identify them, and this is true for humans as well as all other animals. And many of you may have heard of about this, how important the microbes in your gut are, for instance. But what I'm going to do today is I'm going to talk about the context. I'm going to start off by talking about the context of my work, and then a little bit about the research adventures. And so this is a baby Hawaiian bobtail squid, and you can see these are the eyes and the tentacles, obviously, and right here through the transparent mantle is what we call the light organ. And the light organ is a series of tissues that support the growth of a bacterium that makes light for the animal. And I'll tell you in a few minutes why it wants to use the light made by these bacteria. This is a baby and this is an adult squid. And later in my talk, I'll show you the difference in size. so you'll see how different they are. But I wanted to show you how beautiful an animal they are. But what I want to start out with is to start out by telling you, study this and what value it might be in general and to the biomedical community. Okay, so as I mentioned, we have this genomics revolution. And what I mean by a genomics revolution is we're able to sequence the DNA of bacteria, which tells us who they are. And so this has led to this recognition that I mentioned that symbiosis is a general phenomenon. And this person right here is a good friend of mine, Victoria Orphan. She's a professor at Caltech. And her partner made her a t-shirt that said mostly microbe. And I love this t-shirt. So I took a picture of her in the t-shirt. And it's just to illustrate the fact that we now know, for instance, that humans have a very, very high bacterial load associated with them for their whole life that keeps them healthy. And so it turns out that that humans are vertebrates, like fishes and frogs and things. We have a vertebral column. Now, vertebrates are characterized by having what they call 10 organ systems, and that is the respiratory system, the digestive system, the reproductive system, and so on and so forth. Well, it turns out that the microbes are really really deeply embedded in the form and function of all of these organ systems, or at least 8 of the 10 of the vertebrate organ systems. And so it's this interaction with microbes has been with us through evolution and really impacts our biology. Down at the bottom here, I have some statistics. So by cell number, why don't you look at the person next to you? That person is 90% bacterial cells by number. You have 10 to the 13th human cells in your body. You have 10 to the 13th human cells, and you have 10 times as many bacterial cells. If you look at by gene number, how many genes are associated with this, in other words, how many different sorts of things could you encode in these two groups, it's about 1 to 1. That is to say, for every bacterial gene, there's a human gene. And then the gene diversity, because there's one type of human, but there are many, 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 many different types of bacteria you have. There's a huge microbial diversity. And so it's thought that the microbial diversity exceeds that of humans by about 200-fold. So you're pretty much run by your bacteria. So what kinds of effects do they have? Well, this was a paper, I worked on a project with a bunch of people to sort of delve into what we know about this up to this point, and this was 2013. And the work that I'm showing here had been the work of about somewhere between 5 and 10 years. And just, it's extremely recent. And so what has been found, as I mentioned, is that the microbes, particularly the microbes in your gut, affect the behavior of everything. They're responsible, for instance, for the maturation of the immune system. When you're born, you have an immature immune system, and it requires interaction with the microbes, particularly at the beginning, that your mother gives you. They're important for the maintenance of your circulatory system, for your digestive system, and then all kinds of other parts of your body. So this just gives you an idea. Fascinating is it turns out that they're becoming more and more clear that the microbes of your gut are very important for the development and health of your brain. And so things like depression and chronic fatigue syndrome and autism and Alzheimer's are being linked to problems that have gone on or are going on with your microbiota. So it's kind of an important field that we've got here. And what the people are studying who study this is they study the trajectory over development. And so what I'm showing here is I'm showing a baby, and the baby acquires with fidelity each generation a subset of what it interacts with. In other words, when you're born as a baby, of course you're born as a baby. When you're born, what happens is you begin to acquire your microbes from your mother as you pass through the birth canal, and when the baby is breastfed. So what's really interesting about this is that it helps the baby mature, and you do not take in just a random sample of things from the environment. You recognize and allow to persist a very specific human evolved subset of the environment, and that is bacteria that associate with humans. Yeah.
What is trajectory?
A trajectory is a pathway, and so I'm going to show you the pathway through life. So you go from establishment as a baby to maturation. So it turns out that you require your microbes to mature most all of your organs and organ systems. So it's really pretty dramatic what happens here. So if you look at an animal, people raise things like germ-free mice. If you look at a germ-free mouse, it's a mess. It's a total mess. Its organs are not formed properly and so on. It really is a reciprocal dialogue with your microbes that's responsible for lots of the maturation of your tissues. So the next thing that has to happen is once you get in there and you grow and the bacteria induce the development of the host, then you have to come to a stability. And what this means is that the host cannot allow the bacteria to overgrow the tissues, nor can the tissues eliminate the bacteria from the system. So this is a really dynamic balance that has to go on here to make sure. And so what happens in autoimmune disease is lots of times you're turning on yourself, and you're looking at yourself as a foreign material. So basically what happens is you're balancing your immune system with the activities of your microbiota. And then finally, the other question that researchers in this area ask is, what in the world is pathogenesis? In other words, when you get a pathogenic microbe like Vibrio cholera, you also have cholera in you that are not pathogenic. What happens? What is the difference between the two? And so what you want to know, is it like the pathogens speak French and the beneficial microbes speak English? It's a different molecular language, or is it the same language? And so that's one of the other major questions. So one of the things that we now know is that because animals grew up in a microbe-rich environment, many of the things that animals share, many characters of how they interact with microbes. So we share many things with corals, the way corals interact with microbes. There are molecular interactions that we share because they're so old and they were retained over evolutionary time. Another very common feature is most of the time the bacteria reside on the surfaces of your cells. Like, for instance, on your skin, you have 10 to the 11th bacteria that keep your skin healthy. You should never use Purell. Let me tell you. The person who runs the NIH human skin studies of skin microbiota, if you use things like Purell, the Purell will go deeply into your skin and destroy your beneficial microbes. Good old soap and water is what's being, so she's on a tear to get rid of all Purell dispensers around the country, which is almost an impossible job. But the other thing is that almost all of symbiotic interactions with animals and their microbial partners, most of them are what we call horizontally transmitted. That is, they're not passed with the embryo or enter on the egg. They're picked up each generation, which is quite a feat. And there are some animals that do what we call vertical transmission. That is, that they put the symbionts in or on the egg. But in this case, this is horizontally transmitted. Now, the really key and important thing to remember with respect to why we do our work, is that in the vertebrates and in humans, these are extremely complex situations. Really complex. Like I said, you have 10 to the 13th bacteria of thousands of species. How are you ever going to know what's going on there? Well, when biologists are faced with really complex situations of daunting complexity, what they often do is they turn to model systems. They turn to systems. They ask questions like, when evolution had this question, what did it do? What is the diversity of how evolution solved these problems? So I'm showing 3 examples here. One is when the fertilized egg becomes a fully grown human organism. Going from the egg to 10 to the 13th cells. This is a minor miracle. George Clooney is a minor miracle. To go from the egg to George Clooney, in my opinion. But this is really an amazing feat. And likewise, we have around 10 to the 14th synapses in the brain. And trying to understand how those synapses are wired and how they work is a hugely complex thing that the biologists are trying to figure out. And then finally, I'm showing here the topic of my talk, and that is the 10 to the 14th bacteria associated with you. And the thing that's different about the 10 to the 14th here and the 10 to the 14th here is that these are all largely the same genome, whereas these are hundreds if not thousands of genomes that are represented in this 10 to the 14th. Okay, so what are biologists doing with symbiosis? How do we learn about how animals live with bacteria? What are the models that we're using? And so we're exploiting what we call nature's toolkit to give us an idea in simple systems of what are the rules? We want to know what the rules are, how these things work. So what I'm showing here is I'm showing the honeybee. And here's Hydra. This is an organism that only has epithelia. It only has surfaces. It has an outside surface and an inside surface. So it's really simple. host as well as bacteria. This is Drosophila, the fruit fly model. This is the worm model. This is zebrafish. This is a leech. And this is what I'm going to tell you about today. This is the Hawaiian bobtail squid. But just for a second, I'm going to go back to the honeybee. How many of you have heard about colony collapse disorder? Yeah, it's a huge, huge problem. And there are some people who are working on the idea that at least some portion of the colony collapse disorder problem has to do with there being too clean in the hives, with them using antibiotics and things like that. There's a really great scientist in Texas named Nancy Moran who looks at the honeybee, the microbes in the honeybee gut. Now, in the human gut, like anybody sitting here, has several hundred different species of bacteria in your gut. Okay, you have lots and lots and lots. The honeybee has 8. The honeybee has 8. And so what's cool about that is you can hope to know how that honeybee microbiota, how the diversity is achieved and maintained in such a simple consortium of bacteria. It's a very cool system. And hopefully she'll be able to shed light on what's going on with with the collapse disorder, which I think is it's going to be great. So, I want to just say that this looking at model systems is a particularly valuable thing to do. And when I speak to the public, one of the things I like to reinforce is the tremendous value of basic research. Basic research is the driver for applied research. If we don't know what we're dealing with, we can't possibly figure out an application, okay? And the example that I love to give is the field of developmental biology. And so how you get from that egg to George Clooney, right? And what I've got here is actually Ned had done a very nice review for Nature Reviews Microbiology, in which he listed between the years of 1935 and 2007 all of the Nobel Prizes that had been given in developmental biology. This is all of them that had been given in the field of developmental biology. And these are the things that they worked on, which many of you are not biologists, so I wouldn't focus on that. But over here, you'll see that all of them were done on model systems. And so, you have over here the fruit fly and zebrafish. The people who were given the Nobel Prize were people who had worked on very, very simple systems. And so we're hoping that the NIH and the NSF in these very rough times will continue to support basic research, because it really is the driver. But this is what we're trying to do in the study of symbiosis. We're trying to get together a bunch of models that will inform us of how this new frontier works. So what are they? Well, I mentioned them briefly, but you can kind of divide them into a few groups. There are vertebrate models that we can use, although vertebrates are really complex. Vertebrates without their bacteria. So you have a mouse that they call as germ-free, and it has no microbes, and it's kept in a bubble. It's not allowed to interact at all with the natural world, because otherwise it would get infected. And zebrafish is the same way. Also simple consortia, and I gave you the example of honeybee. These are also models that people are using. And then what I'm going to tell you about is a binary system. Now, a binary system is really great because you have tremendous resolution. You always know what to whom in this situation, right? Because you've got one microbial species and you've got one animal host. And so there are a few of them that are available now. And Ned Ruby and I were kind of among the very first to be doing this. And we started this work in 1988 here in Hawaii. I was actually a postdoc at Scripps Oceanography, and Ned was on the faculty at the University of Southern California. And we began this work there and have continued to study this for the last 30 years.
And I've only been here back at Hawaii, at the University of Hawaii for the last 2 years, not even 2 years. Then before that at the University of Wisconsin, so I'm very happy to be back in Hawaii. Okay, so let me tell you a little bit about this gorgeous bobtail squid that you have here in the reefs of the Hawaiian archipelago. So this is Euprymna scolopes, and that's the genus species name of this beautiful animal that lives in the sand flats of the Hawaiian archipelago. And like I said, we study this light organ system to try to understand how bacteria chronically or persistently colonize animal tissue. So if you were to open, anesthetize Euprymna and open its mantle cavity out and lay it on its back, you would see that it has a really complex bilobed organ in the center of its body cavity. To look deep inside of here, you would see that there are bacteria, luminous bacteria, colonizing the apical surfaces of the host epithelia. Now I've actually colored these in, so they don't look like that in a regular transmission electron micrograph. But this was so that you could see the bacteria in relation to the host tissue. This is Vibrio fischeri. It's the genus species of the symbiont that lives in this light organ. And Vibrio fischeri is being taken, this is a plate that's been streaked. So you take the light organ, you grind it up, and you streak it on a bacteriological Petra plate. And what happens is Vibrio fischeri grows up, and you can take a picture of it by its own light. And so that's what's happening here, is you see Vibrio fischeri by its own light here. So what does this animal do with these bacteria, with the light produced by these bacteria? Well, basically, it does a behavior called counterillumination. So this animal is a night-active predator. So Euprymna scolopes comes out at night to forage in the water column. And when it does, imagine hanging in mid-space where you have nowhere to hide. What do animals do when they hang in mid-space and they have nowhere to hide? Well, I remember that last week I think, Angel Yanagihara was here talking about the box jellyfish. What did they look like? Can anybody tell me what it was that allowed them not to be seen in a three-dimensional homogeneous environment? Totally, yeah, very translucent. And so, the light basically largely passes through them. So in these environments that are three-dimensionally homogeneous, there are only a few solutions. You can be transparent if you're really simple, like a jellyfish. You can be silvery, like snorkeling with schools of silvery fishes. They'll turn and you won't be able to see them, and then they'll turn back and they'll flash the sunlight will flash against their silvery sides. So they confuse the predator. The last thing an animal can do, particularly in light fields at night or light fields that are very reproducible, like in the deep oceans, light out of their ventral surfaces of the same color, angular distribution, and intensity of downwelling moonlight and starlight. And in that way, they don't cast a shadow against the visual field of a predator looking up from below. So Euprymna, this little bobtail squid, has a predator, several predators in their environment. One of them is a lizardfish. For those of you who know lizardfish, they sit in the sand and they look up for prey. And as the bobtail squid goes across the visual field, it has a luminescence on, so that the lizardfish has a difficult time seeing that bobtail squid. So it's a camouflage. It's like a Klingon cloaking device or something. So I'm just showing here the way it controls its light emission. So the light organ is embedded in the ink sac of the animal. All cephalopods have ink sacs so they can squirt out ink. And what this ink sac has been modified to do is act as an iris around the glowing culture of bacteria. And so when the light is dim, these shutters or iris closes down, so very little light goes out. Bright, when the moonlight is bright, the shutters or the iris gets bigger to allow more light out to better match the brighter light. So it has control over the bacterial luminescence, which is at a constant level. The other thing that it can do is all luminescent reactions require oxygen. They all are oxidations, and the animal can withhold oxygen. Yes?
I have a question.
Yeah.
If they flash their luminous, the light down there,
Yeah.
how do they sense the moonlight from above?
So it is thought, yeah, so the question is, that's a very good question. If they're flashing their light down, how do they know how much light is coming up? In other words, how do they integrate that information that there's a certain amount of light coming from above and I should put out a certain amount of light out of my ventral surface? There's almost certainly a connection with the eye. And it's thought that there are neural connections. We've never studied this, but there are very, very likely neural connections with the eye. Cephalopods also have something called photosensitive vesicles in their skin. Yeah, and that's another possibility. But with regard to counterillumination, no one's really ever studied that very, very much. Yeah, some of the deep sea squid have been studied, but not much. So here we are. So this is Kewalo Marine Lab, where the action happens, where the research goes on. And what happens, here I am at night. I'm actually, in this particular picture, I'm in Kaneohe Bay, just after dusk, walking around collecting Euprymna from the shallow sand flats. We collect them and we bring them on in from Coconut Island. We bring them on into tanks at Kewalo Marine Lab. And anybody who wants to visit and see the squid, I'd be happy to have you come down. Just write me an email, and I'm there on Fridays. The rest of the time I'm being the director of PBRC up on campus. But we have them in tanks. We go out and we collect the adults, bring the adults back. We only need about 8 females and 10 or so males to keep us in tens of thousands of juveniles a year. Because what happens is they lay big clutches of eggs on rocks, and the females lay a series of clutches. And so they're very easy to maintain and produce quite a few. And so we don't worry about depleting the environment of Euprymna. So here's what I showed you before. Here's the baby, and here's a picture of an adult, and this is the size difference. And so although the babies look like, little adult squid, I mean, they don't have really true larval stages. They have a pretty good size difference there. And so they hatch out of their eggs. It takes them about 20 days to hatch out of their eggs. And then it takes them about 2 to 3 months to become sexually mature, and then they live about 9 months. So as I mentioned in my diagram of the trajectory of life and your microbes, just to refresh your memory, these major questions of harvesting them from the environment. How in the world do you get them? How do you know them? How do your cells recognize the right bacteria? These are the questions that the researchers are asking. What is the influence of the bacteria on host development? How do you make a stable association? And what's going on here? So what do we know about these from the Euprymna system, these questions? Well, firstly, we know that as far as development is concerned, we know the following. What I'm showing here is, I didn't want to spell this out because it's a little arcane, but what happens is the bacteria have on their surfaces something called microbe-associated molecular patterns, MAPS, microbe-associated molecular patterns. And what they are is they're molecules that only microbes have. And so when an animal sees one of these molecules, it says, oh my gosh, there's a microbe. Okay, I recognize that. And they're recognized, these microbe-associated molecular patterns are recognized in animals by what is called PRRs, or pattern recognition receptors. So how many of you have heard of endotoxin? Endotoxic shock, toxic shock, septic shock. Anybody? Yeah. So those are all due to a MAMP in bacteria, a MAMP called lipopolysaccharide. And it's a cell surface molecule on gram-negative bacteria. And that particular molecule has been thought of historically as a toxin. But biologists are having to change their whole attitude toward this kind of thing, because it turns out that all microbes, all gram-negative bacteria have LPS. They all have that particular MAMP. And so it can't possibly be a toxin. So as I mentioned earlier on, it was like, is pathogenesis French and beneficial like English? Or is it the same language that is used in a different way? Well, it turns out that MAMPs, it is the same language used in a different way. It's like the difference between a civil discussion and an argument. So a civil discussion being a beneficial association and an argument being a pathogenesis. So the molecules, same molecules used in a different way. And so we found this about 10 years ago in the squid, that the squid's development is driven by this molecule. Okay? The squid's light organ development, the development of its organ. So I thought, oh, maybe that only happens in the squid. But we showed that bacteria could induce animal development. So I'm over in Paris at a meeting of the Pasteur Institute. And I thought, I was giving the opening talk. And I sat down after my talk, and I was very jet lagged because I was coming from Hawaii. And I sat down and, excuse me. The guy who got up right after me is a researcher at the Pasteur. And he said, so I'm sitting there and I look up and the squid is still on the screen. And I'm thinking, what? I'm really hallucinating. I'm really jet lagged. Because this guy studies mice. So it had long been known that mice respond, their gut develops in response to gram-negative bacteria. And so he said to himself, well, if a squid can do it, maybe a mouse can do it. And indeed, the exact same molecule drives development on the mouse. Excuse me, guys. So very highly conserved over evolutionary time. So as I've mentioned a little bit, this idea of mutualism versus pathogenesis, we found a whole host of things that are shared. Virulence factors, what have been called virulence factors and have been called host defenses, are just the language that animals and bacteria use to talk to one another. Now, I'm going to finish off by telling you one very specific story. And that is, everybody loves circadian rhythms. And so I'm going to talk about how this association becomes stable. And at the basis of the stability is a really dynamic circadian rhythm. That is a rhythm that goes over the day-night. So what we did was we asked the question of whether or not the language, the molecules in the two partners change over the day-night cycle. In other words, is their language different over the day-night cycle? When the animal buries, so the animal is out at night, right? In the morning, it buries in the sand. And so when it buries in the sand, it vents 95% of its bacteria into the surrounding environment, and then they grow back up. Okay, and so what we did was we looked right before that venting and right after that venting, and we see huge changes in the language that between the two partners in the transcriptional regulation of these two partners. And what we saw by looking at this more closely at the tissues, we saw that what happens is the tissue undergoes a tremendous remodeling on the day-night cycle. So not only is the molecular language different, but the tissues are different at these different times of day. And so what are the bacteria doing in response to this? So one of the things that the bacteria does, so what happens is the bacteria, the animal's membranes fall apart. So what the bacteria do in response to that is they turn on all the genes to eat membranes. And then the animal completely repolarizes its epithelia. And in other words, it looks really nice again. It's not all disrupted and there aren't a whole bunch of membranes around. And so the bacteria turn all those genes off. That's what you see here in red. And then they begin to turn on genes associated with the use of another nutrient that the animal provides. And that's the animal, I'll tell them, for those of you who are biologists, that is chitin. The animal provides chitin to the bacteria. And so they go on a day-night cycle. The bacterial, there's a rhythm in the bacterial metabolism that's in response to what the animal is providing it. So it has a breakfast and it has a dinner, and they're different, and it's on this circadian rhythm. So what drives this rhythm? You know, how does this happen? How does the animal know? And what cues of Vibrio fischeri, what cues might Vibrio fischeri use to entrain rhythms of these scolopies? So actually, I should mention that this work was done by a really talented student of mine, Elizabeth Heath Heckman, who's now a postdoc at Berkeley. And she worked on this project to find out what's underlying this rhythm. Well, what she did was she studied a gene called cryptochrome. And cryptochromes are molecules that are sensitive to blue light. And the bacteria make blue light. Okay? And so there was some suggestion that the activity of these might be involved in this rhythm. And one of the major reasons was cryptochromes are clock genes. So everybody in this room has at least 2 clock genes, one in your head and one in your gut. Okay? And so Elizabeth began to study these clock genes, and they drive your circadian rhythms. This is your central clock, and this is your peripheral clock. Okay? So she started to study this. And what you see here are the vertebrate ones. Here's vertebrate cry. You know, you have 4 actually. 1 through 3 and 4. And the invertebrates typically have 2. They have cry 1 and cry 2. And just, you know, Euprymna had 1 in its head and 1 in its light organ. Okay? So what was cool about that was she asked the question of whether or not what is cry 1 and cry 2 regulated in the head. And what she found was that in the head, ES cry 1 is not very drastically regulated. But ES cry 2 is regulated in the head. Whereas in the light organ, ES cry 1 is regulated and ES cry 2 is not. So that was further proof that ES cry 2 is the one in the squid's head, and ES cry 1 is the one in its light organ. And they're cycling on a day to night. That gene that encodes that protein that sees blue light goes on and off and on and off in a cycle. The thing that's cool about this is the luminescence, because it's used at night, is 12 hours off. In other words, the brightness of the bacterial light is 12 hours off the brightness of the environmental light that's up in the head. So the cry of that's coming in from the environment and being sensed, cry 2, cycles with environmental light 12 hours off of the cycling with the cryptochrome 1 in the light organ. So isn't that cool? Just want to show, this is the tissue, this is where the bacteria are. The bacteria here are in red. So if you took, this is a histological section showing the bacteria in the animal tissue. The blue is the animal tissue. And Elizabeth made an antibody, which is a molecule that allows you to localize where is that cryptochrome relative to those bacteria. And it's right on the edge of the host cells. So it right interfaces very closely. And so it's in a place where it makes sense. Elizabeth also showed that bacteria are required for the cycling of the gene in the light organ. So Cry1, if you don't have the symbiont, Cry1 does not cycle. And then to top it all off, Ned Ruby's lab gave us a mutant in light production. So that bacterium was perfectly fine every other way, except it couldn't make light. And if you gave the animal that, you would not get cycling. If you gave the animal the Lux mutant over here, it looked like a non-symbiotic animal. Not only did it require that the bacteria be present, but it required that the bacteria be luminous. And what she was able to do by shining light underneath was she was able to show that she could complement that problem in that defective strain that didn't make light by giving the animal light from underneath at the same periodicity that it would see naturally. And then she could restore. I should say that light alone did not work. It requires the presence of the bacteria, and it requires bacterial MAPs. So why is this important? Well, I've shown you these things about the circadian rhythm of Euprymna. Well, it turns out that they now know that the bacteria are running your circadian rhythms. The reason why you get jet lagged, the reason why all these things are happening are because your circadian rhythms are disturbed. One of the things that they found is that one of those MAMPs, which is a molecule of the cell wall of bacteria, gets into your bloodstream, goes up to your brain, and trains the third wave of the mammalian sleep cycle. And in rats, where they were able to cure them of their microbiota, they found that their sleep cycles are a total mess. And then if they inject this molecule into the circulatory system and it goes to the brain, they can restore the third wave of the mammalian sleep cycle. So this is one of the reasons why a lot of people feel that when you take antibiotics, your sleep cycles get messed up. So it's a very important, this understanding of how intimately associated the bacteria are is a really big field now. And it's just this particular area. So I would go to meetings. We've been studying this since 1998, and we would go to meetings and we'd say, you know what, you guys have to look at circadian rhythms. You have to look at circadian rhythms. And they did everything. They showed circadian differences in everything in the gut, like the gut epithelium and the gut immune system and everything. They never looked at the bacteria until about 4 years ago. And they went, oh my God, not only are they on a rhythm, but they're setting the rhythm. So it's pretty important. So I'm going to finish there, and I'm going to just say thank you to Ned, who's sitting quietly out there, for partnership all these years in studying the squid vibrio system. And this is back in Madison 3 years ago, before we moved back to Hawaii, Madison, Wisconsin. And there are now something like, is it 16 labs, Ned, working on the squid vibrio system, our academic children and grandchildren across the country. And some of them were able to come and celebrate with us the 25th anniversary of the study of the squid vibrio system. So with that, I will say mahalo, and I'll take any questions. Yes?
When you said
this is really simple, but I don't understand a lot of this. But when it expels,
Yeah.
you said it expels the bacteria.
Yeah.
Where do they go?
So this is a really interesting question. So they expel them into the mantle cavity, into the body cavity of the squid, and when the squid ventilates, they get sent out into the surrounding seawater. Now the interesting thing about this is that microbiologists early on in describing the bacteria of Hawaii said Vibrio fischeri did not occur here. And one of the major reasons, there are a couple of reasons, but one of the major reasons is that they typically look offshore. Vibrio fischeri is principally around where the squid host is. The squid host came from Japan down the Hawaiian archipelago, and the only place it hasn't been found is on the Big Island. And maybe there's not a habitat there for it yet, but Vibrio fischeri seems to follow them. And when the babies are born, if you take water that has adults in it, it's very infective. The farther you move away from adult populations, if you put a baby in that water, the less likely that baby will get colonized. It takes it a long time and may never get colonized. And so there's a bit of habitat loyalty to the whole situation. But the bacteria go out into the surrounding water, and in an environment like, we often collect in Pico, right down in the Ute Valley area in Malama. And in that area, we think that the bacteria are somewhere between 50 to a couple thousand cells per mil. Now that's against a background of 1 million bacteria per square centimeter per mil of seawater. And so they're not all that abundant. But what's there is very likely to have been a squid symbiote.
Can I have a second part of that?
Yeah.
How do they rebuild it? The bacteria just reproduce fast enough that they get enough for the next cycle?
So that's that. When
they expel them, now they need more.
Oh, actually, no. I should have been more precise with that. They expel 95%. What's remaining behind grows up to fill the light organ each day. And so what happens when they're babies? Is on each side of the organ there are 3 independent crypt spaces that will be colonized. And when they're babies, it looks like each one of those crypts may get a single bacterial strain, or a few. Is that right? Yeah. And so that what happens is you get a single bacterium in each one of these crypt spaces. So what that suggests is that a single bacterium, no matter how many are in the seawater, a single bacterium will make its way all the way to that baby crypt and grow out and fill the organ. Yes?
Thank you very much for the talk. So you mentioned, what's the benefit of the chitin to the bacteria?
Yeah. So Vibrios as a genus of bacterium, Vibrio cholera, Vibrio perihemolyticus, Vibrio harvii, Vibrio fischeri, are chitin degrading bacteria. And so they feed on chitin. And they have a series of genes that enable them to transport they export chitinases that break down chitin, and then they have genes that are associated with helping them transport it into their cells, and then they have chitin utilization genes. And so it's a characteristic of them, and it's a nutrient.
So basically, so the host produces that and they feed off it and then produce something like that. So that's the mechanism.
Yeah, at least at night. During the morning, they seem to feed on the host membranes. Yeah, on the day-night cycle. So first it's right at dawn, the microvilli, the edge of the cells, they just fall apart into that crypt space where the bacteria are, and the bacteria begin to grow. They turn on the genes associated with growing on membranes. And then as soon as there's no more membrane left, the animal begins to feed them chitin.
Right, because they get to critical mass and everything. Great, thank you.
Yeah. Yes.
Let's say I have eyes that can see at night and I can detect them.
Yeah.
At what depth would I'm swimming in the water.
Yeah.
At what depth would I see the bobtail squid? That's the first question. And secondly, during the daytime when they go into the sand, do they have a optimum depth of sand that they like to go to, or do they go to any sand level?
Yeah. So in answer to the first question, when we go collecting them, my favorite place to collect them is in that much water. So I just walk along the edge, and you can see them really well if they're sitting on the sand. Oftentimes, when they're not swimming around, they'll sit down, they'll sit on the top of the sand, and then you can just scoop them up. But they've also been found as deep as 100 meters, this particular species. So they do go deep, and we're finding, and we've always found, the entire time we've been fishing, that it's very difficult to find them in the summer. And the shallow sand flats get extremely hot in the summer, and so we think they move offshore. And then in the winter, but they breed all year round. And so when they're just a little bit harder to find in the summer, but when we find them, they breed and lay eggs, not a problem. Yeah, and so, yeah. What was the other question? Oh, how deep do they go? How deep do they bury? They bury, it's amazing. So we'll have this much sand in their condominiums in the lab. They have each of them has their own condominium. And they will have just an inch or an inch and a half of sand, and they will bury so well that in a condominium this big, with a base that big, people will look in there and you can't see them. You cannot see them. You can't find them because they're such good barriers. Yeah.
They have that rhythm going, and what happens if you disrupt that? You mentioned take antibiotics or something, and people, we lose it. And what happens to that animal if you disrupt that cycle?
Yeah. So these guys, they don't like it at all. If they're symbiotic, they don't like it at all. In fact, if you hold them in the dark, the adults particularly become very physiologically stressed. And so we've never carried those experiments very far because it's kind of a difficult thing to study. In the juveniles, what happens is if you hold them and don't let them vent, they just hold it and hold it and hold it and hold it and hold it, and then they become incontinent. They just start to leak out cells. They just get to the point where it's just too much. But certainly you've disrupted their cycles. And one of the interesting things is there's a maturation of the animal. It's such that the juveniles are quite different. They're up in the plankton up until, they're hanging around mainly in the plankton until they're about 3 or 3.5 weeks old. And then they become fully nocturnal.